What?
What did you say?
I said if you know everything that I said during the review, you should do very well in the
There's no other
final.
reviews.
Okay.
All right. So let me move along because I have to get through a bunch of stuff, and I ran out of time a little bit in the other class. So, chapter 4 was where we left off. That's the tissue chapter. It says describe the 4 basic tissues, examples of each type, and the cells found in each. So, what are the 4 basic tissue types? Connective, epithelial, muscle, and nervous. All right. Epithelial tissue. Where do you find that? Yeah, it's on the surface of things. Usually, it could be the outer surface, like your skin, or the inner surface, like lining your hollow organs. Epithelial tissue is usually avascular. What does that mean? No blood vessels. And remember, it's all about cells, right? Epithelial tissue has lots of cells packed together and almost no matrix in between them, right? Very little extracellular matrix. Remember, epithelial tissue can be classified based on how the cell looks and then how many layers there are. For example, what would you call it if you saw a single layer of flat cells?
Squamous, simple squamous.
Simple squamous, right? What if you saw a layer of tall, thin cells that looked like it was multiple layers, but it was really only one layer? Pseudostratified, and if it's tall, thin, it would be, what's that shape called? Columnar. So that'd be pseudostratified columnar. It looks like it's multiple layers because of the position of the nuclei, but if you look closely, all the cells actually touch the basement membrane. They just don't all reach like the free surface. Connective tissue was the broadest group. There's a lot of subtypes that fit in there. 2 of the most common cell types that you see in connective tissue are fibroblasts. They make collagen fibers, and then macrophages, which are kind of immune defense cells that are hanging out that can gobble up bacteria and things like that. Remember that connective tissue often sits underneath the epithelial tissue, right? You'll have an epithelial tissue on top, then you'll have something called the basement membrane, and then underneath that, you'll have the connective tissue. And we did talk about some different membrane types in this chapter. We talked about serous membranes, which we also talked about in the first chapter. That's like the pleura, the pericardium, the peritoneum. We talked about cutaneous membrane, bless you, which is just a fancy way of saying the skin. And then we also talked about mucous membranes, right? These are like lining the inside of your GI tract or lining the inside of your respiratory tract. When you have a mucous membrane, there was a special name for the connective tissue layer of a mucous membrane. Anyone remember what that special name was? It was abbreviation. You said it. What was it? Lamina propria. Good job. Yep, lamina propria. Different types of connective tissue. Blood's a connective tissue, for example. What are the cell types that you find in blood? What kind of cells do you have in blood? Red blood cells. What else? White blood cells. What are the little tiny things that kind of, what are those? Platelets? Yeah, you have platelets in blood. I will tell you on the final that they use the words neutrophils and lymphocytes. That's just, those are subtypes of white blood cells. I don't know why they're pulling those terms out yet because we really don't cover it until you get into A&P2. But if you see the words neutrophils and lymphocytes, those are just specific types of white blood cells, okay? Another type of connective tissue are the 2 dense types of connective tissue, dense regular and dense irregular. Now, the dense part means that there's lots of collagen fibers. What can you say about dense regular connective tissue? What does that mean about the collagen fibers in dense
Same
regular?
direction,
They're all running parallel in the same direction. What about dense irregular?
different direction.
They're all running around in different directions, okay? All right, moving right along. Muscle tissue. How many types of muscle tissue are there? 3. What are they?
Skeletal,
Cardiac, smooth, and skeletal, right? We'll talk about them in more detail in the muscle chapter, which we'll get to in a second. And then lastly, nervous tissue. True or false? The only types of cells in nervous tissue are neurons.
False.
False. What's the other cells called that help out? Yeah, neuroglia or glial cells, right? Know the locations and functions of all tissues outlined in class. Well, that's pretty broad, so let me just tell you a few things. If I said keratinized stratified squamous epithelium, where would you find keratinized stratified squamous epithelium in your body?
Skin.
In your skin, like the epidermis of your skin is that. If I said, oh, I don't know, pseudostratified ciliated columnar epithelium with goblet cells, where would you find that? That'd be like in your respiratory tract, so inside your nose or in your trachea. If I said simple columnar epithelium with microvilli. Where would you find that? That's mostly in the lining of your gut, like in the lining of your stomach or the lining of your small intestine.
What did you call that again?
It was microvilli.
No, the term that was the location, the simple. What was it?
Simple columnar epithelium with microvilli. Yeah, that would be mostly like in the small intestine where you find that. Simple cuboidal epithelium. Many glands have simple cuboidal epithelium. The tubules of the kidneys have simple cuboidal epithelium. What else? I don't know. There's a whole bunch, but that's good enough. Know the locations of the types of cell junctions. So cell junctions were ways that you hold cells directly to each other. And we talked about, if you remember, we talked about something called adherens junctions, desmosomes, hemidesmosomes, gap junctions. Those were some examples. What are the ones called that hold cells together really closely so that no fluid can leak between the cells? Those are tight junctions, right? And you find those like in the bladder or like in the stomach. Any place like where you want to hold a fluid and you don't want it to be able to leak out, you usually have like those tight junctions, okay? Next it says describe glands. Unicellular, multicellular, merocrine, apocrine, holocrine, which is most common. So first of all, remember there's two types of glands in general in the body. What's the name of the glands that have ducts? Those are called duct glands. No, they're called exocrine glands. If you have a duct, you're an exocrine gland. If you don't have a duct, what kind of gland are you?
Endocrine.
Endocrine, okay? Now, in AMP1, we mostly talk about exocrine glands, all right? It's not until you get into AMP2 that we talk about endocrine glands. And one of the big chapters where we talk about exocrine glands is actually the next chapter, the skin chapter, because we talk about sweat glands and sebaceous glands. Those are all examples of exocrine glands, right? Now, exocrine glands, you should know, they have ducts. They're usually multicellular. They're made up of multiple cells. Although we did talk about one exocrine gland that's unicellular, that kind of looks like a wine container. What were those cells called? Goblet cells. Remember, that's an example of a unicellular exocrine gland. Most exocrine glands, though, are multicellular. And then the words merocrine, apocrine, and holocrine, those are words that describe how the exocrine gland secretes the material, okay? And anyone remember what happens if you use the merocrine method? How do they secrete with the merocrine method? Don't all answer at once. All right. Basically merocrine, first of all, you should know merocrine is the most common method. And with merocrine, you're just doing exocytosis. You're putting whatever you want into a vesicle, and then you're releasing that vesicle outside the cell. So it's just exocytosis, okay? That's the most common method. What happened if you use the apocrine method of secretion? Anyone remember what gland cells do when they use the apocrine method? The apocrine method is when it's kind of like the cell loses its head. It like breaks off and it becomes the secretion, and then the cell has to regrow its head, okay? And then what happened with the holocrine method?
We kill everyone, right?
The whole cell dies and it becomes the secretion, okay? All right, next we had the integumentary system chapter, which is a fancy way of saying what? Skin. Describe excuse me. Describe the structure and function of the skin and its exocrine glands. Fine. So, first of all, let's talk about the function of the skin. What were some of the functions of the skin?
Protect.
Protection, temperature regulation. What else? What's the vitamin you can make in your skin?
D.
Vitamin D, excretion of certain waste products. The dermis can act like a blood reservoir. Those are some of the functions. Sensation, sure. In terms of the structure of the skin, what's the outermost layer of the skin called? The epidermis. And then the layer below that is called the dermis. What's the layer below that called?
Subcutaneous.
Subcutaneous layer. Is that considered part of your skin?
No.
No. All right. What was the other name for the subcutaneous layer? Hypodermis, right? Now, in terms of the glands, you have two types of glands in your skin. You have sweat glands and you have oil glands. Oil glands are also known as sebaceous glands. Of your sweat glands, there were two types. What's the name of the sweat glands that you find all over the body? I think you said it. What'd you say? Yeah, eccrine sweat glands are found all over the body. They have a simple coiled tubular shape to them, and they're every place in the body. What's the type of sweat glands that you only find like in your armpits or the groin area? Those are apocrine sweat glands, okay? Now, oil glands, which are also known as sebaceous glands, they always empty into what? Hair follicles, okay? So that's something to be aware of. Differentiate between thick and thin skin. So where on your body do you find thick skin? Palms of your hands, soles of your feet. Where do you find thin skin? Everywhere else. Which type of skin has hair?
Thin.
Thin, right? Thick skin doesn't have hair. Which type has more sweat glands, thick or thin?
Thick.
Thick, right? Think about like when you're nervous, they talk about sweaty palms, right? Thin skin doesn't have as many sweat glands. What about sensory receptors? Who has more sensory receptors, thick or thin? Thick also, right? Think how sensitive like your fingertips are, your, et cetera. And then lastly, which one, well not lastly, but which one has oil glands?
Thin.
Thin, right? Remember, oil glands always have to empty into a hair follicle. So if you don't have hair follicles, then you also won't have oil glands, right? So thick skin doesn't have oil glands. And then epidermal ridges, that's a fancy name basically for fingerprints or toe prints. You're gonna only see that in thick skin, okay? Know the order of the epidermal strata, cells in each strata. So assuming that we're in thick skin, how many layers do you have of the epidermis do you have in thick skin? 5. How many layers do you have in thin skin? 4. So let's assume we're in thick skin, going from outside to in. What's the outermost strata called? Stratum corneum. Then you have the stratum lucidum. Then you have the stratum granulosum. Then you have the stratum spinosum. And then you have the stratum basale, right? Which is the layer that's missing in thin skin?
Lucidum.
Lucidum. All right. Now, it says know the cells in each strata. So remember, in the epidermis, you have a couple of different types of cells. What's the most common cell that you have in the epidermis?
Keratin.
Keratinocytes, right? And they're the guys that make keratin, which is this tough protein that's on the outside of your skin. But there are other cell types too. What's the ones called that make pigment? Melanocytes, right? They make melanin. What's the purpose of melanin?
Check from the sun.
It absorbs UV light so that it doesn't harm the DNA of your cells, right? Melanocytes are found in the stratum basale. What other special cell type is found in the stratum basale? What's that? I don't know, Merkel cells, right? The special sensory cells, they're found in the stratum basale. And then the other type of cell that plays a role in your immunity is the what? Langerhans cells. They're usually found someplace in the stratum spinosum. Okay. Describe and know the orders of the phases of deep wound healing. So if you have a deep wound what does that mean? Like how deep did that go in your skin if you have a deep wound? It means it at least got into the dermis, right? It means it's gone all the way through the epidermis and it's at least gotten into the dermis. It might have even gone deeper, but at least got into the dermis. And when that happens, you got to go through 4 phases of deep wound healing. What's the first phase? That's the cleanup phase, which is called the inflammatory phase, okay? During inflammation, blood vessels to the area will dilate. That will increase blood flow to the area. And then the walls of the blood vessels sort of separate and become more permeable so that white blood cells and things that are in your blood can squeeze out through the wall and then get into the tissue and kind of clean up any bacteria, destroy any bacteria things, just kind of get it cleaned up. And then you can actually go into the other steps. What's the second step after the inflammatory phase? That's called the migratory phase. And it's called that because epidermal cells undergo mitosis and they sort of migrate their way across to kind of close the epidermis, okay? At the same time that they're fixing the epidermis, in the dermis you have fibroblasts laying down collagen fibers to kind of repair what's in the dermal layer, right? Phase 3 is called the proliferative phase. That's basically just more of the same stuff that was happening during the migratory phase. And then phase 4 is the maturation phase, and that's when that can take weeks to months. That's when the blood vessels kind of become normalized. When you first have a wound, the scar will look pretty red, but over weeks to months, it starts to look more white as the blood vessels become normalized. All right, chapter 6 is the bone tissue chapter. It says, describe the histology of bone, cell types, differentiation. So going back to chapter 4, which of the four main tissue types is bone classified as? Connective, right? So if you look at bone, you can either look at the cells that are in the bone, or you can look at that space outside of the cells called the extracellular matrix. Let's look at the cells first. The cells there's several different types. 3 of the types were kind of related to each other, and the 4th type was sort of an oddball. The osteogenic cells, those are bone stem cells that can differentiate and go on to become what? What do osteogenic cells become? They become osteoblasts. What do osteoblasts do? They build bone. Now, I will tell you on the final, it doesn't say build bone. Instead, it says it in a really obscure way. It says secrete bone matrix. Whatever.
Oh, nice.
That's so lame,
We
right?
can come and ask you about that, right?
You can come and ask me. I'm just telling you about it now. That's fine. Secrete bone matrix is just another way.
For every point.
It's just another way of saying build bone. All right. Now, when the osteoblasts build bone, they eventually surround themselves completely, and then they can't really build anymore, and they go into sort of semi-retirement, and they become what cells? Osteocytes, right? So osteogenic cells become osteoblasts, which then become osteocytes. Those 3 are related. What's the 4th group that was kind of an oddball? Osteoclasts. And what do? They break down bone. Now, I will tell you, on the final, they don't say it that way. On the final, they say resorb bone matrix. Whatever. It means to break down bone. Okay?
Does the science community talk like this?
Yeah, some do. Some try to keep it real, but there's a lot of
Big
snobs
bang
out there.
theory
They're
stuff, like-
elitists. They figure like they spent all this money on their education, they might as well talk like it, right? So they're kind of they're losers. Let's just face it. All right. So those are those cells. Now there are some other cells to be aware of. When you're an embryo and your skeleton is first developing for the first time, there's an early type of connective tissue cell called a mesenchymal cell. And those are kind of the first cells to get together, and they will differentiate and become sort of the bone cells. But they're an early type of connective tissue cell that you pretty much just see in the embryo. Those are mesenchymal cells. Another type of cell you should know are chondrocytes. What are chondrocytes? Those are mature, not bone cells, but mature, what's the rubbery stuff that you have at the front of your nose? Those are mature cartilage cells. Remember, anytime you see chondro, think cartilage. Okay, so chondrocytes are mature cartilage cells. Describe the hormones involved in calcium homeostasis. CT, PTH, calcitriol, estrogen, IGFs. So I know for a fact some of this stuff we didn't cover, or at least not in as much detail as they cover in the final. So let me go through this. First of all, do you guys know what, in this context, what does CT stand for? No. No, that's just what he said.
Cartilage?
No, so in this context, and it's kind of dumb, I would have thought connective tissue too. It actually stands for calcitonin, which is a hormone, okay? You can, listen, come up during the final. If there's ever an abbreviation you don't understand, come up and I'll tell you what they mean, okay? Don't struggle. I told you about that one year when I didn't tell the students what ACH meant. And yeah, that girl came on and was, ah, and I'm like, what are you talking about? All right, anyway. What does PTH stand for?
Parathyroid.
Parathyroid hormone. And then does anyone know what IGF stand for? Those stand for insulin-like growth factors. And I'll tell you what they do here in a second. Insulin-like growth factors. So first of all, let's talk about calcitonin, or they're breathing at CT. What does calcitonin do to blood calcium levels? Increase it or decrease it? Lowers it, it decreases it, okay? Anyone remember the memory trick? Yeah, calcitonin tones down blood calcium levels. And then you just have to remember that parathyroid hormone does the opposite. It increases blood calcium levels. Now, on the final, instead of using the word blood, instead they use the word serum. It means the same thing as blood.
You have never used that.
I know I haven't. I'm just trying to keep it real. It's basically a synonym for blood. Serum, calcium levels, right? It means the same thing.
Why don't you put that on the board?
Don't
Okay,
freak out.
for serum.
Can you
Yeah.
put everything on the board?
No,
Make
just
it-
come up with no,
Why not?
because it's a slippery slope. There's so many little weird words that they use, and it'll just take forever. Just come up and ask me. I think you'll get it. You'll get it from the context. I really don't think it'll be that confusing. And there's really not that many. But if you have a question, just come up and
Remember
ask me.
this moment in time when you see syrup going.
Yeah. So when you see serum, it means blood. All right. So calcitonin decreases serum calcium levels, and parathyroid increases serum calcium levels. What's calcitriol? It's just a name for the active form of vitamin D. All right, the vitamin D that you get in your skin is not the final version. It has to actually go to your liver, then it has to go to your kidneys, and after it goes to your kidneys, it gets made into this calcitriol. It's the active form of vitamin D. What does vitamin D even do? What's the purpose of vitamin? Once you have the active form of vitamin D, what does it do? What does it do? It does strengthen your bone, but how?
Something in the GI tract.
Yes. So calcitriol goes to your intestines, and it helps you absorb calcium from the foods that you eat, right? You could eat foods that have a lot of calcium in it, but if you don't have enough vitamin D in your body, you'll just poop all that calcium out and you won't get it in your body, all right? Estrogen, why the hell are we talking about estrogen now? I will tell you why we're talking about it. So first of all, when I say estrogen, what do you usually think of?
Female.
You think of like female hormone, right? Okay. Well, the truth of the matter is, both men and women have some estrogen in them. Estrogen and testosterone structurally are very similar, and it's really easy to convert testosterone into estrogen. Now, obviously, men don't have as much estrogen as women do, but they do have a little bit. In both sexes, and I know I didn't really emphasize this in class, but I'm going to say it now. In both sexes, estrogen plays a role in your bones in telling your epiphyseal plate when to close. So estrogen is kind of controlling when you stop growing, right? Remember when you're like, you know, in your late teenage years, your early 20s, your epiphyseal plate, which was made out of cartilage, is going to turn into bone, and then you stop growing. Estrogen is kind of what controls that, okay? And then IGFs. I know I probably didn't talk about these either, but let me explain what they do. IGFs stand for insulin-like growth factors, and they help you grow. They're sort of the middleman between growth hormone and your bones, all right? You have this hormone in your pituitary called growth hormone. You'll learn more about it in AP2. Growth hormone is the main hormone that causes you to grow. And when you're a child or a teenager, the pituitary produces growth hormone, and then it makes you grow. But growth hormone doesn't directly go to your bones and make you grow. Instead, growth hormone goes to your liver. It makes the liver release these things called IGFs, and then they go to your bones and actually make you grow. So IGFs are like the middlemen in terms of making you grow.
So is that what differentiates people's ability to grow? Is like the liver's output of this IGF?
It could be
Or
one
is it
factor.
the
It could be
third
one
factor?
factor. It could also be how much growth hormone their pituitary releases in the first place. It could be how sensitive their bone cells are to the effects. So there could be a number of things. It could be. It could be. All right. All right. Chapter 9 is the joint chapter. It says, describe the types of joints, know examples of each, and the movements permitted by each. So there were 3 structural classifications and 3 functional classifications for joints. If you have a joint capsule, be careful. If you have a joint capsule,
You would not have wanted that.
what type of a joint are you called if you have a joint capsule? You're a synovial joint. Remember, a joint capsule means that you're a synovial joint. If you don't have a joint capsule, then you have to ask what type of tissue is holding the bones together. If it's dense connective tissue, then that's going to be called a fibrous joint. If it's cartilage, then it's going to be called a cartilaginous joint. Okay? So those are your 3 choices. You're either a synovial joint, a fibrous joint, or a cartilaginous joint. For function, there were also 3 choices. What's it called if you have a joint where the bones don't move? That's a synarthrosis. What if the bones move a little bit? No. Amphiarthrosis if they move a little bit. And then what if they're freely movable? Diarthrosis, right? What should you know about this? Know that, you know, most of your famous joints that you think of when somebody says joint, like your knee or your elbow or shoulder or whatever, most of those guys are going to be classified as synovial joints structurally, and functionally they're going to be classified as diarthroses. Okay? Describe synovial fluid and the types of movement at synovial joints. What's the purpose of synovial fluid? Yeah, it helps the knee move more, or it helps any joint move more smoothly. It's like a lubricator inside of the joint cavity that helps it move more smoothly. Also, don't forget, it also has an important job in delivering oxygen and nutrients to the cartilage that sits at the ends of your long bones, that articular cartilage, because that cartilage doesn't have blood vessels in it to deliver oxygen and nutrients, so it sort of relies on the synovial fluid to do that. Types of movement. These were the words that you learned, which hopefully you remember these words. What's it called when you move a body part away from the midline? Yeah, abduction or abduction, right? What's it called when you move a body part toward the midline? Adduction or adduction. What's it called when you decrease the angle between bones? Flexion. When you increase the angle and it brings you back to the anatomical position, that's called extension. And if you go beyond the anatomical position, that's hyperextension, right? What's it called if you turn your forearms so that your palms are facing anteriorly, like this? That's supination. And what if you turn it so they're facing posteriorly? That's pronation. Good. What's it called if you spin a bone or you move a bone around its own longitudinal axis like this?
Rotation.
That's rotation. What's it called if you do a combination of flexion, extension, adduction, abduction like this? I'm hearing cracking. Yeah, that's called circumduction, right? What's it called when you bring your thumb across to touch your other fingers? Opposition. What's it called when you bend your ankle, kind of like if you're standing on your heels? Like, what would that movement at my ankle be called? That's dorsiflexion. And what if I was standing like on my toes, and what would that be called? That's plantar flexion. All right. Chapter 10 was the muscle tissue chapter. That's all I'm saying. Yep. Differentiate between the three types of muscle tissue and then the anatomy of the sarcomere.
Squall.
What was that? Oh, it's snow. Wow.
Like a mouse or something.
I know. Was that reaction to the snow?
I
Yeah.
thought it was probably like the next subject.
All
Do
right.
we have to know about neobards?
You have to know everything.
Yes. Yes. I only will say yes to those questions. I never answered, do you need to know? Sorry. All I will say is what I said before. If you study what I go over in this review and know it like the back of your hand, you should do very well. All right. Differentiate with the three types of muscle tissue, anatomy, and sarcomere. So what were the three types of muscle again? Skeletal, smooth, and cardiac muscle, right? So let's talk about skeletal muscle first. Skeletal muscle, if you look at it under the microscope, how does skeletal muscle look under the microscope? It's got stripes, which are called striations. What else? How many nuclei does one skeletal muscle cell have? Multiple, right? It could be like 100 nuclei in one skeletal muscle cell. Remember, skeletal muscle cells, or any muscle cells, are also known as muscle fibers. Skeletal muscle fibers can be really long. They run parallel to each other. And then what can you say about the control? Can you voluntarily control skeletal muscle? Yeah. Yes, it's connected up to your somatic nervous system and you can voluntarily control it. All right, cardiac muscle. Does cardiac muscle have stripes?
Yeah.
Yes. What is it that cardiac muscle and skeletal muscle have that make them have stripes?
Oh, sorry.
They have sarcomeres. Sarcomeres are the ways that the thick and the thin filaments are organized, and that's what gives them the stripes. Smooth muscle still has all the usual stuff, the thick, the thin filaments, the actin, the myosin. They're just not arranged in sarcomeres, so smooth muscle doesn't have stripes, okay? Other things about cardiac muscle, probably the most characteristic thing about cardiac muscle are the little lines that you can see where one cardiac muscle cell connects with another. What are those lines called?
Intercalated disc.
Intercalated discs, okay? That's any other type. And then smooth muscle, as I said, doesn't have stripes. Smooth muscle cells look kind of fat in the middle and then they taper at the end. That's called a spindle shape, okay? Regarding cardiac and smooth muscle, can you voluntarily control them?
No.
No, they're involuntary, and that's because they're connected up to your autonomic nervous system, right? In terms of the anatomy of the sarcomere, this is when you had all those dumb letters that represented different parts of the sarcomere. Remember that? What was the names for the discs that you had at the beginning and the end of each sarcomere? Those were the Z discs. The Z discs are attachment points for actin, and that's where the thin filaments are attached. In the very middle of the sarcomere, you have something called the M line, and that's an attachment point for the thick filaments. You have something called the A band, What was the A band? Anyone remember? The A band was the full length of the thick filament, period. The I band, this is so painful. The I band was the thin filament that didn't overlap with any thick filament. And the H band was just the opposite. The H band was the thick filament that didn't overlap with any thin filament. I'm glad I don't have to take this. All right. Sequence, next it says sequence the events of skeletal muscle contraction. All right, so once again, I was super lazy and I didn't want to rewrite this, so I already put this on the board, but I'm going to talk about it before I show it off. So remember, skeletal muscle cells, they always have to be connected up with somatic motor neurons in order, they'll never just contract on their own. They always got to have the motor neuron, tell them to contract. And a motor neuron, a somatic motor neuron, is always going to release acetylcholine. Acetylcholine will cross the synaptic cleft at the neuromuscular junction, and it will bind to acetylcholine receptors that are on the muscle cell. And remember, those acetylcholine receptors are essentially ion channels for sodium. And when acetylcholine attaches, these sodium channels, or they're also known as ligand-gated sodium channels, they're going to open up, and that's going to allow sodium to rush in. Sodium is going to rush in, and you're going to basically create an action potential that travels down the length of the muscle cell. And at first, the electrical signal at first is going to be on the outside of the muscle cell. But do you remember, what was the name of the structures that allow the electrical signal to go deep inside the muscle cell? Yes, thank you. Those are called T-tubules, all right? So as the electrical signal travels, it will go and follow into those T-tubules. The T-tubules bring the signal into the muscle cell, and sitting right next to the T-tubules were these little structures that, in other body cells, they're kind of like endoplasmic reticulum, but we have a special name for them in muscle cells. They were called sarcoplasmic reticulum. When the electrical signal goes through the T-tubule, it passes by a sarcoplasmic reticulum, and it causes the sarcoplasmic reticulum to release what ion into the sarcoplasm?
Calcium.
Calcium. Calcium gets released, and how does calcium actually cause the muscle to contract? What does calcium do that makes the muscle to contract?
Binds.
It opens up the binding sites on the thin filaments, all right? The thin filaments are made up mostly of actin, but then there's these two other proteins that kind of block the binding sites called troponin and tropomyosin. When calcium's released, it binds to the troponin, which then unblocks the binding sites so that the thick filament can then attach to the thin filament. And then assuming that you also, what else besides calcium, what else is the other thing you need to make a muscle cell contract?
ATP.
Assuming you have ATP, then the actual contraction will begin, okay? So here is what basically that in a nutshell. Okay, so I will leave that up there. You can look at it, take a picture of it. I don't care. Now, the other thing I want to mention about skeletal muscle contraction is what happens when you stimulate a muscle cell to contract. Oh, and I didn't put my top on this. Hold on a second. I know, this is like rocking my world. Hold on, let me figure out what's a good one here.
Oh, Valentina, black class.
What you laughing at?
The Last Supper.
Getting attacked again.
Alright, so that's actually what I was trying to do over here. So here's what would happen if you stimulate a muscle cell with a motor neuron, and you could create this thing called a myogram where you basically monitor what happens. So if you stimulate a muscle cell once with a motor neuron, you get something called a twitch contraction, where the muscle cell contracts and then it relaxes. Okay? Now, what would happen if I stimulate that muscle cell again a second time? You basically get a second stronger contraction. So if I waited, so I stimulated once there, but then let's say I stimulated again at this point, what would happen is more calcium would be released into the sarcoplasm, and instead of going and fully relaxing, you then get a second stronger contraction, and that's known as wave summation. Now, if I kept doing that and just kept waiting, if I allowed it to relax a little bit, but then stimulated again, you'd get something that would look like this, and that was referred to, maybe you remember, as unfused tetanus. Versus if I stimulate it even faster, if I don't wait until it relaxes a little bit, like if I stimulate it actually while it's still in the contraction phase like this, then I would get a line that looks more like this, okay? And that was called fused tetanus, okay? So the point is, when you stimulate a muscle cell multiple times, it will build and give you stronger and stronger contractions, okay? Now, there is something to know, though, about, actually, no, that comes up later. Forget that. When the thick and thin filaments attach to each other and they start pulling, then you go through this thing called the contraction cycle. I showed you a picture of that. And there were 4 steps to the contraction cycle. What was the name for the step when the actual pulling takes place? When the thick filament pulls the thin filaments along, there was a specific name for that step. That was the power stroke. All right, I think it was like step 2 on that diagram that I showed you guys. Now, the way that they worded on the final is the power stroke is the rotation of cross bridges to generate force.
Good.
I know. It's the pulling step. That's what it is. All right.
We'll be able to clarify that. We'll be able to clarify it during the test, right?
You can clarify it. Just come up and say to me, like, does that mean the pulling step? And I'll be like, yes, that means the pulling step. The power stroke is the rotation of cross bridges to generate force. It's just a really snooty way of saying it's the pulling step. All right. Describe the order of the reactions which produce ATP and skeletal muscle. So what this is referring to is, remember, where a muscle gets its ATP from depends on how long you're using the muscle for. If you're just running a sprint or doing some activity that lasts for like 10 seconds, where do you get the ATP from? Something called creatine phosphate, okay? If you're using your muscles for maybe up to a minute, but no longer than a minute, you can use what's called anaerobic respiration, or it's sometimes called glycolysis, anaerobic glycolysis. They use both names. But if you're going to run like a marathon that's lasting for several minutes to hours, then you have to get the ATP from what's called aerobic respiration. Okay. All right, chapter 12, nervous tissue. Differentiate between the neuron and neuroglia. So neurons, remember, they're famous, they're big, they get to send electrical signals. Neuroglia are small, they don't send electrical signals. They do actually outnumber the neurons, remember, by like as much as 25 to 1, so there are more neuroglia. But they do other important stuff, they just don't send electrical signals. So let's talk about the neuroglia then in terms of their types and what they do. Schwann cells and oligodendrocytes were related to each other because they both do what? What do Schwann cells and oligodendrocytes do? They make myelin. Where do the Schwann cells do it? In the peripheral nervous system. Where do the oligodendrocytes do it? In the central nervous system, right? Some more type of neuroglia. Ependymal cells. What did ependymal cells do? They make, I'll give you a hint, it's a clear fluid that goes around.
CSF.
They make the CSF, okay? Ependymal cells make the CSF. We had another type of cell called a astrocyte. What did the astrocyte do? It makes the blank, blank, blank. Blood-brain barrier, right? Remember the blood-brain barrier? And then microglia. Those are cells that kind of act like macrophages. They're there as defense cells. They can gobble up bacteria and things like that. Describe an action potential and graded potentials, EPSPs and IPSPs. So we're going to go through this. In the next section, we'll go through the events of an action potential. I'll talk about that. Just one thing I want to mention is, what's the name for that thing that, like, when a neuron is not doing anything, it has that electrical difference across its cell membrane? What's that called? The resting membrane potential. And what's that value? -70 millivolts. All right. that means that the inside of the neuron is slightly negative compared with the outside of the neuron. There were 3 reasons why that existed. Number 1, there are more, I'm not going to go through all the details here, but there were more potassium leak channels than sodium leak channels. Number 2, there's a lot of big anions that are stuck inside the cell that can't get out, and anions have a negative charge to them. And then number 3, the sodium-potassium pump. Remember, the sodium-potassium pump pumps 3 sodiums out, but only 2 potassiums in, and that creates a difference in the charge there too. Something else to say about graded potentials and action potentials. With graded potentials, the size of a graded potential depends on what? How strong the stimulus is. So a graded potential is, the strength of a graded potential is based on the size of the stimulus. Remember, graded potentials come in 2 types. If you have a graded potential that goes up like that, that's called it goes by a couple of names. That's called a depolarizing graded potential, or it's also called an excitatory graded potential. Or if you were looking at a postsynaptic neuron, they call that an EPSP. All of those things mean the same thing. Depolarizing graded potential, excitatory graded potential, or EPSP, they all mean the same thing. And they're due to some stimulus causing a stimulus-gated sodium channel open. On the other hand, if you have a stimulus that causes a stimulus-gated potassium channel open, then you get a graded potential that looks like this. That's called a hyperpolarizing graded potential, or it's also called an inhibitory graded potential, or if you're talking specifically about the postsynaptic neuron, they call that an IPSP. Okay? The size of the graded potentials depends on how strong the stimulus is, right? For example, if somebody just touched you lightly on the arm, maybe you'd get a graded potential that's that big, right? If somebody pressed a little bit more firmly, you'd get one that was like that big. And then if somebody really pressed firmly, really strongly, you'd get something that looks like that. So the size of a graded potential depends on how strong the stimulus is. Does the size of an action potential depend on how strong the stimulus is?
No.
No. What's that principle that action potentials follow? All or none. What that means is you either trigger an action potential or you don't. But when you do, they always come in the same size. You don't have different size action potentials. And that's one thing, all right? Another thing you should know about action potentials and graded potentials is graded potentials have the ability to add together to make bigger graded potentials. What's that property called? Summation. Okay? Can action potentials do summation?
No.
No, they can't do summation because they have a what? They have a refractory period, okay? And they actually have 2 types of refractory periods. What's the name of the refractory period where no matter how strong or how strong the stimulus is, you can't get another action potential? That's the absolute refractory period. And then what's the refractory period called where, yes, you can get one, but you have to have a stronger than normal stimulus? That's the relative refractory period, okay? So action potentials have those refractory periods, so they have to wait, so they can't add together and do summation the way that graded potentials can, all right? Next it says sequence the events of an action potential. So this will be another sequence section on the final, and I'm going to walk you through this and then I'll show you what I wrote on the board here. Basically, what they're going to do on this question is they're going to give you kind of a starting point, and then you're going to have to follow it through from there. And the starting point for this question is going to start, it's going to assume that you have 2 neurons. Okay? Again, this would be the synapse, right, where they're meeting. What's the name of the neuron that comes before the synapse?
Presynaptic.
That's the presynaptic neuron, and then the one that comes after it
Post.
is the postsynaptic neuron. So on this question, they're going to assume that you're actually at the synaptic end bulbs of the presynaptic neuron. In other words, you're there. All right, so what happens when, let's suppose you're in that presynaptic neuron. What happens when the action potential gets to the end of the presynaptic neuron? What is it going to do? It's going to release neurotransmitter, right? And do you remember what actually happens in the synaptic end bulb to cause the neurotransmitter to be released? there was something that had to rush into that calcium, all right? In the synaptic end bulb, right there are voltage-gated calcium channels. When calcium rushes in, that will cause the presynaptic neuron to release its neurotransmitter into the synaptic cleft. The neurotransmitter is going to cross the synaptic cleft, and it's going to bind to a receptor for that neurotransmitter that's on the dendrites or possibly the cell body of the postsynaptic neuron. And that receptor is usually going to be like some kind of a ligand-gated channel. It'll be, it could be separate structures, or it might be an all-in-one deal, such that when the neurotransmitter binds, it will open up that channel, and then it will allow an ion to move through that channel, right? Now, if we assume that it's a ligand-gated sodium channel, if you open up a ligand-gated sodium channel, which way is the sodium going to move? Into the neuron or out of the neuron? Into the neuron, okay? Remember, when a neuron's at rest, or when any body cell is at rest, there's lots and lots of sodium outside of your body cells and not that much sodium inside. So if you open up a ligand-gated sodium channel due to a neurotransmitter binding to it, sodium's going to rush in, and that's when you're going to get a depolarizing graded potential like this one, okay? Obviously, if the stimulus caused a ligand-gated potassium channel open, then you would get this, and it'd be a different situation. But for this, and on the final, they're going to assume that it was a ligand-gated sodium channel that was open, and so you're going to end up getting a depolarizing graded potential, or since we're specifically talking about the postsynaptic neuron, you could call it an EPSP. It means the same thing. Now, one EPSP, or one graded potential, is probably not enough to reach threshold on its own. What's the value for threshold?
-55.
-55. One EPSP is probably not enough. However, EPSPs, or graded potentials, whatever the hell you want to call them, they can add together, they can do summation. So, assuming that they add together and then you reach threshold, what will you get if let's say you hit threshold perfectly, what are you going to get?
1.
One action potential. What if you didn't quite hit threshold? What's going to happen?
None.
Nothing. It stops. And what if you go above and beyond the threshold?
Multiple.
You get multiple action potentials, right? Now, when it comes to the action potential, there's two main phases to an action potential. The phase when you're going up going up like that, that's called the depolarizing phase. And that's due to the opening up, if you remember, of voltage-gated sodium channels, right? It all starts on the axon. What was the spot on the axon that sits kind of close to the cell body where it all began?
Yeah.
That was the trigger zone. Remember that? So it's all going to start at the trigger zone. The first voltage-gated sodium channel opens. Sodium rushes in, which then causes the next one in line to open up. Sodium rushes in there, which then causes the next one to open up. And it's kind of like you're hitting this series of dominoes down the length of the axon. Eventually, though, then you'll gradually have the opening of voltage-gated potassium channels. Potassium rushes out, and as it rushes out, you enter into the repolarizing phase when you go down and become negative again, okay? So it always starts with the depolarizing phase, and then it goes into the repolarizing phase. So with that introduction, and because I'm too lazy, let me show you what I had written. It's basically this, which is basically covered in that.
Sorry.
Right? I feel like I've got like a professional cameraman here. I feel like you should have a much more impressive camera
I know,
than just
right?
that. Like you should have some big thing over your shoulder like up there. I'll leave this up here at the end too if you want to get another shot if you want to get a good shot. All right. Hold on. I gotta yell at somebody. For a second. Not like literally, but figuratively. All right.
It's pretty easy, right?
This looks really horrible.
It's just packed.
Yeah.
It's like, I don't even know if you have like a lung or
It's not just hip, there's other professors, so maybe they'll be a little bit more
They're
lenient
jerks.
Sounds like they're more complicated.
All right. All right. Next chapter was spinal cord and spinal nerves. It says, describe the anatomy of the spinal cord and spinal nerves, the opening for the spinal nerves, and then meninges. So first of all, anatomy of the spinal cord. Where does the spinal cord anatomically begin? The foramen magnum. And then where does it end?
The something CM.
Where?
Starts
Well,
with
conus
C.
medullaris, yeah,
Yeah.
but I mean, it's like what vertebrae level?
Oh.
Like around L2 or right about there, okay? If you look at the spinal cord from like the back, it's going to be a little bit thicker in the cervical and lumbar areas. Those are called the enlargements. Why are they bigger in those areas?
Is this
Extra neurons are coming in from either your arms or from your legs, right? So they're going to be bigger in those areas. Now, the spinal cord is attached to the spinal nerves. What division of the nervous system is the spinal cord in? Central. What division are the spinal nerves? Peripheral. Good. Remember that the spinal nerves are considered mixed nerves. What does that mean?
They have sensory and motor.
They have sensory and motor neurons in them. Good. The way that the spinal nerves attach to the spinal cord is usually in these things called roots, right? And you had a dorsal root and a ventral root. How can you tell the dorsal root from the ventral root? Ganglia, right? That's like this little bulge that's on the dorsal root that you don't see on the ventral root. What kind of neurons do you find in the dorsal root? Sensory. What kind of neurons do you find in the ventral root? Motor. Good. Remember, as the spinal nerves come out of the spinal cord, they have to leave the vertebrae. What's the name for those holes that are on the sides of the vertebrae? Those are the intervertebral foramen. Good. All right. And then talking about the topic of meninges. Meninges are these kind of protective coverings that you have around the spinal cord and also around the brain. What's the innermost meninge called? Yeah, that's called the pia mater. What's the middle layer called? Arachnoid mater. And then the outer layer is called the dura mater. Now, I will tell you, I'll even write, I already wrote it on the board actually. On the final, some snob decided, I don't know if you look it up in some dictionary, the singular form of meninge, which I always just thought was meninge, I guess it's technically meninx. Whatever.
I know they want us to do well.
I don't know. They're hating on you for some reason. I have no idea why. All right. Anyway, that means meninge. Don't stress about it. Come up and ask me if you need to. All right.
We're gonna need a button.
No.
Yeah.
All right. Where was I here? Differentiate between a nucleus and a ganglion. So, with both of those things, you have a group of neuron cell bodies sitting together someplace. Okay? Now, if you're in the central nervous system, what are you called? That's a nucleus. So a grouping together of neuron cell bodies in the central nervous system is called a nucleus. Versus a ganglion or a ganglia, that's a grouping together of neuron cell bodies in the peripheral nervous system. Okay? Now, just to make your life miserable, they don't use the word grouping together or clustered together. They use the word aggregate. That means the same thing as a grouping together. Or an aggregate is just a cluster or a grouping together or something. Okay?
Reverse ball.
I didn't make this
Were
final.
you the one?
I was not the one. No, I would tell you. I wouldn't do that. I keep it real. No, the person that did these complex, she actually just retired last year. She's probably laughing all the way.
Can you call her back and ask
All the way
her
to the
why?
bank. She's laughing all the way to the bank.
She's a really sweet lady, and she makes
I
us
bet.
good food. So yeah. But she's tough when she loved the nervous system. That was her baby. So the tougher she could make it, all the better. She loved being detailed and super tough about it.
Somebody nuggets.
Yeah. Yep.
She's a sweet lady.
She's a sweet lady, but she would just torture you relentlessly with the nervous system. Describe the tracts of the spinal cord and the functions of each.
More?
That's actually going to come up in chapter 16, so I'm going to skip that for now. Okay. Describe the steps of the reflex arc and the spinal reflexes. Discuss contralateral, ipsilateral, intersegmental. So, how many steps are there in a reflex arc? 5. What's the first part of a reflex arc? That's the sensory receptor, right? What's the second part? That's the sensory neuron. What's the third part? Yeah, the integrating center or the control center, they sometimes call it. What's the fourth part? That's the motor neuron, and then what's the fifth part?
Effector.
The effector. If it's a somatic reflex, what's the effector always going to be?
Skeletal.
Skeletal muscle. If it's a autonomic reflex, it's going to be cardiac smooth or glands, right? Now, in terms of the specific reflexes that we discussed, we basically just went through 4. We talked about stretch reflexes, tendon reflexes, flexor withdrawal reflexes, and then the crossed extensor reflex. Those are the 4 that we talked about. 3 of those 4 are ipsilateral reflexes, meaning that the sensory signal comes in on the same side that the motor signal goes out. Which one was a contralateral reflex, though?
It's a cross, right?
The crossed extensor, right? That was kind of a weird one. It went to the other side. What else can I tell you about these reflexes? Some of the reflexes are intersegmental reflexes. That just means that when the sensory neuron comes in to the spinal cord, branches are given off to involve as many levels of the spinal cord as possible. The reason why you do that is it will then lead to a stronger response because you'll be getting more levels involved, okay? That's called an intersegmental reflex. And the flexor withdrawal reflex and the crossed extensor reflex are actually both examples of intersegmental reflexes. If you go back and look at the diagram in that PowerPoint, you can see what I mean, how once the sensory signal goes in, it's kind of getting a whole bunch of spinal cord levels involved. Describe a nerve plexus, the 4 plexuses discussed, and the phrenic nerve. So nerve plexuses are ways of organizing neurons in order to send them to different areas of the body, okay? Specifically, the plexuses that we discussed here were made up of the anterior rami of your spinal nerves, all right? So when the spinal nerves, just to refresh your memory, when the spinal nerves come out through the intervertebral foramen, they immediately start giving off branches. And the biggest branch is the anterior ramus, but then there's a posterior ramus, and there's a meningeal branch, and there's the rami communicantes, etc. But anyway, it's the anterior rami that will form plexuses. Now, do all anterior rami form plexuses? No. What area of the spinal cord did you not have a plexus in? Thoracic. Okay? That's really the exception to the rule. Everywhere else, you're going to have a plexus. What was the most superior plexus that you had? Cervical plexus. And what was the famous nerve that came off the cervical plexus? The phrenic nerve. Why is it famous? Yeah, it helps you breathe by going to the diaphragm. And what was the memory trick for it? C3, C4, C5 keep you alive because those are the spinal nerves that contribute to it. All right. What's the next plexus you would have after the cervical plexus? Brachial plexus. That's in charge of organizing the nerves that go to your upper extremity. Okay. Then you get a break in the thoracic area. No plexus in the thoracic area. Then the next plexus you encounter is the lumbar plexus. What's the famous nerve that comes off of the lumbar plexus?
Femoral?
Femoral nerve. Good. I know somebody wanted to say sciatic. Don't say it. Then you come to the sacral plexus. What's the famous nerve that comes out of the sacral? That's the sciatic. And then finally you have this little coccygeal plexus. All right. Now you may have noticed on the study guide they say the 4 plexuses discussed. We actually just listed 5. The reason why they say 4 is because remember some textbooks will actually combine the sacral and the coccygeal into one and they'll call it just the sacrococcygeal. All right. So don't worry about that. It's not going to be an issue on the final, but I'm just explaining it to you. All right, chapter 14, brain and cranial nerves. Describe the functions of the basal nuclei, BBB, pituitary gland, and CSF. So the basal nuclei, remember, are those bits of gray matter that are in the cerebrum that are deep inside the cerebrum, which is a little bit weird because most of the gray matter in the cerebrum is on the outside as that cerebral cortex, right? So deep inside, though, you find the basal nuclei. What do the basal nuclei do? They help with subconscious contraction of skeletal muscle, like swinging your arms automatically when you walk. They help with that. They help you start and stop body movements. And then they also help in other ways not related to movements, like they help regulate emotional behaviors. They can affect your attention and memory and planning. So the basal nuclei are kind of a big deal. They do a lot of different things. The BBB, what does that stand for?
Blood-brain barrier.
Blood-brain barrier. It's basically there to prevent potentially harmful things in your blood from getting into your brain tissue. It's made up of the tight junctions of the cells that line the walls of the blood vessels. So the blood vessels are made up of cells in the wall called endothelial cells. Those endothelial cells in most places in your body are not held too tightly together, so it's fairly easy for substances in your blood to kind of squeeze their way through the wall of the blood vessel and get in your tissue. But in the brain, it's a lot harder because those cells are held together with tight junctions. That's number one. Number two, you have astrocytes, which help surround the outside of the blood vessel, and that provides another layer that, again, all of this makes it very difficult for substances that are in the blood from being able to get through and get into your brain tissue. Pituitary gland is a little gland that hangs underneath your hypothalamus. The two of them are closely related. The hypothalamus has direct axons connecting it to the pituitary. The hypothalamus has neurons in it, so it's considered part of your nervous system. But it actually also has cells that produce hormones, so the hypothalamus is also considered a part of what other organ system? Endocrine, okay? So it's considered as being a member of both organ systems. CSF, what does that stand for? Cerebrospinal fluid. Where is it made?
In the arachnoid.
It's made deep inside your brain, inside of the ventricles at spots called choroid plexuses. It's made by ependymal cells that filter your blood, and they create the CSF. The CSF flows through the ventricles on the inside of your brain. It goes through that cerebral aqueduct. It eventually gets into the fourth ventricle. Some of the CSF will continue down in the central canal of the spinal cord. Some of it, though, will go out through little holes and it'll get into the subarachnoid space. Then it circulates around the outside of your brain and spinal cord until finally, where does it ultimately go?
Arachnoid.
It goes into the arachnoid villi that return it back to the blood. Okay. Define decussation and projection tract. Actually, I think some people asked me about the word decussate during lecture exam 4. What does the word decussate mean? It means crossing or crossing over, okay? It's just a spot where neurons can cross sides. You have a pyramidal decussation in the pyramids of the medulla. So kind of on the front of the medulla are these bumps called pyramids, and there's a pyramidal decussation. That's a possible crossing over. Another place, it's a little confusing. They'll use the word decussation as a crossing over spot, and it literally means crossing. But another place where neurons cross sides are at the commissures, right? So you talk about the anterior white commissure in the spinal cord or the gray commissure. Those are also crossing over spots. Or even the corpus callosum, that's considered a commissure. Okay. Projection tracts, those are tracts that go up or go down. If they're going up, what kind of signals are they carrying? Sensory. If they're going down, they're carrying motor, right? Know the number, name, and function of the 12 pairs of cranial nerves. Which cranial nerve has its own dermatome? Which cranial nerves are mixed? Which are special sensory? So, first of all, remember that the cranial nerves are part of your peripheral nervous system. Okay. Cranial nerves 1, 2, and 8 are all special sensory, right? 1 and 2 do olfaction and vision, and then 8 does hearing and equilibrium. Cranial nerves 3, 4, and 6 are related because they all move the eyeball. Cranial nerves 5, 7, 9, and 10, those are the mixed cranial nerves. What does it mean to be mixed again? It mean they have sensory and motor function to them. Cranial nerve 5 and 7 have some similarities because they affect your face. Remember, cranial nerve number 5 is the trigeminal. That does sensory to your face. So the answer to which cranial nerve has its own dermatome, that's cranial nerve number 5, all right? It's covering your face. Remember, most dermatomes on your body are handled by the spinal nerves, but actually your trigeminal has a dermatome and it's the one on your face. Cranial nerve number 7 is the facial nerve, and that's the big one that does the muscles of your face. Now it also does taste and stuff, but it's probably best known for doing the muscles of your face. Which cranial nerve is known as the wanderer? The number 10, the vagus. It's called that because it goes really far in the body. It doesn't just stay up in your head area like most of the cranial nerves do. It can go down as far as like your stomach, your intestines. It goes really far away. It covers sensations from internal organs. It can monitor blood pressure. It can monitor blood gas levels like oxygen and carbon dioxide levels in the blood. It can send signals to your heart and make it beat more slowly or send signals to other places. Chapter 15 was the autonomic nervous system, differentiate between somatic and autonomic nervous system. So somatic nervous system, remember, are you consciously aware of the sensations with the somatic nervous system? Yes. Can you voluntarily control the motor function of the somatic? Yes, and that's because it always goes to skeletal muscle. Are you consciously aware of the sensations in the autonomic nervous system? No. And can you voluntarily control the motor function? No, it's involuntary, and that's because it's going to smooth muscle, cardiac muscle, or glands. Now, the motor division of the autonomic is divided into the sympathetic and parasympathetic divisions. Sorry, give me 1 second here. All right, where was I? All right, differentiate between sympathetic and parasympathetic division of the autonomic nervous system. So first of all, where does the what's the nickname for the sympathetic? Fight or flight. What's the nickname for the parasympathetic? Rise and digest. Where does the sympathetic division originate, or what's the other nickname for the sympathetic? Thoracolumbar. It begins in either the thoracic or the lumbar spinal cord. Where does the parasympathetic begin? Craniosacral. So it's either beginning in your brainstem or in the sacral part of your spinal cord. Let's go back to the sympathetic for a moment. Sympathetic division is the one that uses the rami communicantes in order to connect it up with your spinal nerves. And remember, there's a white rami and a gray rami. Why is the white rami white? It has myelinated axons in it. Why is the gray rami red? It has unmyelinated axons in it. When you turn on your sympathetic system, what happens to the size of your pupils? They get bigger. What happens to salivation? It decreases. What happens to your heart rate? Increases. What happens to the diameter of your airways? They dilate. All right, good enough. Now let's talk about the parasympathetic. So the parasympathetic is also called craniosacral. What were the 4 cranial nerves that have parasympathetic branches in them? 3, 7, 9, and 10 have parasympathetic branches in them. That's going to be coming off of like the brainstem. When you're in the sacral part of the spinal cord, those cranial nerves give rise to what are called the pelvic splanchnic nerves. Okay, and some body changes with the parasympathetic. What happens to the heart rate with parasympathetic? Slows down. What happens to your pupils? They get smaller. What happens to like stomach movement and acid secretion in the stomach? It increases, right? All right. Differentiated between preganglionic and postganglionic neurons, cholinergic neurons, nicotinic and muscarinic receptors, and adrenergic neurons, alpha and beta receptors. So remember, the stupid autonomic nervous system is a 2-neuron system. It takes 2 motor neurons to go from the central nervous system to reach the effector, which makes it a pain in the butt. The first neuron is called the preganglionic neuron. The preganglionic neuron is going to have its cell body someplace in the central nervous system. Preganglionic neurons have axons that are myelinated, and preganglionic neurons always release acetylcholine. Okay? Postganglionic neurons are going to have, on their dendrites and cell bodies, obviously they have to have a receptor for the acetylcholine that's being released by the preganglionic neuron, and they're going to have what are called nicotinic acetylcholine receptors located there. The postganglionic neurons themselves, they're going to have their cell body outside of the central nervous system. And the type of neurotransmitter the postganglionic neuron releases is going to depend really on what division you're in. If you're in the parasympathetic division, postganglionic parasympathetics always release acetylcholine. If you're in the sympathetic, it's more complicated. Sympathetic postganglionics usually release norepinephrine unless they're going to sweat glands, in which case they release acetylcholine. When you have a effector, it's going to have a receptor type that matches up with the neurotransmitter that's being released. So if norepinephrine is being released, the effector is going to have a norepinephrine type receptor on it, or what's known as a adrenergic receptor. And there were 2 categories of norepinephrine receptors. They were called alpha and beta, and then there were like alpha 1, alpha 2, beta 1, beta 2, beta 3. If you have a situation where acetylcholine is being released, then the effector is going to have a muscarinic type of acetylcholine receptor on it. Okay? Okay, fine. Describe, chapter 16, sensory, motor, and integrative. Describe the structural classification of sensory receptors. So here we're talking mostly about like the simple types of receptors you find like in your skin, for example. And there were 3 types of structural classifications. The simplest type would be just to have the bare dendrites of the sensory neuron act as the receptor, and those were referred to as free nerve endings. If you wrap the free nerve endings with connective tissue, those were called encapsulated nerve endings, and those would be things like your Meissner's corpuscle, your Pacinian corpuscle. And then the third option is to have an entirely separate cell act as the receptor. In other words, it's not part of the sensory neuron, it's separate from it. And a good example of that would be like Merkel cells, or even like gustatory cells with your sense of taste. They're separate from the sensory neuron. Describe the somatic sensory pathways and the motor pathways. So you got sensory pathways and you got motor pathways. And again, sensory pathways are always going to be going up, or they're also called ascending pathways. And then motor pathways are also going to be going down, or they're called descending pathways. Now, let's just talk about the sensory for the moment. And there's 3 pathways here that they basically want you to know. The posterior column medial lemniscus pathway, the anterolateral / spinal thalamic pathway, and the trigeminal thalamic pathway. Let me discuss these. First of all, the posterior column medial lemniscus, they usually abbreviate that the PCML pathway. And that basically refers to those tracts that you find in the posterior columns of the spinal cord. If you remember, there were basically 2 tracts there called the gracile fasciculus and the cuneate fasciculus. Anyway, what should you know about the PCML pathway? It does touch. It also does things like conscious proprioception, okay? So that's the PCML pathway. And the PCML pathway is basically covering most of your body except for your head, okay? The spinothalamic, or the anterolateral pathway, whatever you want to call it, that's covering the same regions of the body as the PCML. So it's doing most of your body except for your face, except it's doing different sensations. The PCML does touch, conscious proprioception, etc., whereas the spinothalamic is most famous for doing pain. It also does temperature, itch, tickle sensations, etc. Okay? So it takes those 2 sensory pathways to basically cover sensations on your body. If you want to talk about sensations on your face, there's one pathway that covers all the sensations on your face, and that's the trigeminal thalamic pathway, okay? And so, you know, touch, pain, itch, tickle, all of those sensations on your face, or inside of your nasal cavity, or inside of your mouth, or even like on your teeth, they're all covered by the trigeminal thalamic tract. So those are all sensory. Let's talk motor now. So for motor, you have what are called direct motor pathways and indirect motor pathways. Direct motor pathways control voluntary movements. Indirect motor pathways control involuntary, sort of subconscious movements, okay? The direct motor pathways, they always start in the cerebral cortex, so their names will always start with the word cortico. The indirect pathways always start someplace in the brainstem, so the beginning of their tracks is going to have some crazy name that represents some place in the brainstem, like tectospinal, or vestibulospinal, or rubrospinal, okay? In terms of the direct pathways, you had the corticospinal pathways and you had the corticobulbar pathways. The corticospinal pathways did voluntary movements of your body. The corticobulbar pathway did voluntary movements of your face. For the indirect pathways, there's going to be 5 separate tracks. They all start someplace in the brainstem, and those included things like the rubrospinal tract, the medial and lateral reticulospinal tracts, the vestibulospinal tract, and then the tectospinal tract. I think I covered all of them. The tectospinal tract is called that because it starts in the tectum of your midbrain, right? Remember, that's where you had like the corpora quadrigemina. So it's starting, actually it starts specifically on the superior colliculi, and then it goes down and then someplace in your spinal cord. Again, the indirect pathways is controlling more automatic things like moving your arms, swinging your arms when you walk, those kind of things. All right, last but not least, chapter 17, the special senses. Describe the anatomy and functions of the eyes, ears, nose, and tongue. So The eyeball basically has 3 layers to it. The outermost layer is sometimes called the fibrous tunic. It's mostly made up of the sclera, which is the white part of your eye, except when you get in the front, and that's where you have the cornea, which is the transparent part. The middle layer is mostly made up of the choroid, which contains a lot of melanocytes in it. It has a dark black color to it. The middle layer is also made up of the ciliary body. The ciliary body is the spot where you secrete aqueous humor. The ciliary body is also that area where you have the ciliary muscle, which controls the shape of your lens by pulling on little strings. Another thing you find in the middle layer is your iris, which is the colored part of your eye, right? And then the innermost layer of your eye is going to be the retina. And, you know, there's a pigmented retina that has a lot of melanocytes in it. But the retina part that we looked at more was called the sensory retina. That's where you have your photoreceptors, your rods and your cones. That's where you had bipolar cells and ganglion cells, all that stuff. If you were to take a pointer, hopefully a soft one, and if you could push the pointer straight through your eye, through the front of your eye until it hit your retina on the back, it should hit a spot called the macula lutea. And in the middle of the macula lutea is a spot called the fovea. The fovea is where you get your sharpest color vision because it has a high amount of cones there, okay? Let's talk about the anatomy of the ear. 3 parts of the ear. There's the external ear, the middle ear, and then the inner ear. The external ear has the auricle, or the pinna, on the outside. Then it has the external auditory canal. And then it stops at what? Your tympanic membrane, your eardrum. Then when you get on the other side of that, you're in the middle ear, which is basically a cavity. Attached to the eardrum on the inside is the malleus, which forms a joint with the incus, which forms a joint with the stapes, which then covers over the oval window. Also connecting into the middle ear is the auditory tube, or the eustachian tube, right? When you get into the inner ear, you have 3 areas: the vestibule area and the semicircular canals. Those areas handle equilibrium, versus the cochlear area handles hearing. If you look at the cochlea, there are 3 kind of tubes inside of the cochlea. The cochlear duct is one of the tubes. That's also known as the scala media. And then you have these 2 other tubes called the scala tympani and the scala vestibuli. Anatomy of the nose, you have what's called the olfactory epithelium kind of on the roof of your nose. That's where you find olfactory sensory neurons, support cells, and basal cells. The anatomy of the tongue, you have all these bumps all over your tongue called papillae. The biggest papillae are the valate papillae that are at the back. The ones that are scattered all over that look kind of like mushrooms are called what? Fungiform papillae. The papillae that are on the sides of your tongue that kind of look like stripes, they're called what? Foliate papillae. And then the papillae that are the smallest that are scattered all over your tongue are called filiform. What was special about filiform papillae? They don't have any taste buds, right? Describe the cells involved in the special sensory pathways and into what areas of the cerebrum do they project. So when you talk about special senses, if you didn't notice, you probably did notice, there are a lot of what are called hair cells involved. Like when you look with your sense of smell or your sense of taste, you had like olfactory hair cells and gustatory hair cells. And if you look really close on those hair cells, there's actually little chemoreceptors that respond to chemicals in the air or chemicals in your food. You also had hair cells involved in hearing and equilibrium, though, remember? Like in that organ at Cordy, there were hair cells, or if you look on the crista, there were hair cells. And in those cases, with hearing and equilibrium, the hair cells are actually getting pulled. Remember, you were tugging on them, and then that created an electrical signal. Another type of cell that you see commonly are bipolar cells. Remember, olfactory sensory neurons are essentially bipolar cells. You also saw bipolar cells in your retina connecting up the photoreceptors with your ganglion cells. Sorry, gotta put something in something. Okay.
Yeah,
Quiet.
because our process is more
The
complex
Drew
than
said it.
12,
The reality of it. If you know it, you know it. How'd you almost fall? That's what I really need to ask.
Backpack cord.
be the backpack.
Five.
that takes you out.
It'll
Oh,
throw
it's
it.
5%. Oh, the quiz is 1%.
Yeah, the quizzes are 1% and they add up to 5%. So we have 5 then, and I didn't go, but I don't know. I would be at 8 too, but I want to get but I don't know. I have to actually
did great,
go
don't worry.
on it. But
You're so.
I don't, I
You're
just
such
don't
a
want
dad.
to
All right.
Yeah, so.
Lastly, it says, know which sensory pathways synapse in the thalamus. It'd probably be easier just to create a list of the ones that don't synapse in the thalamus, in which case the answer would be smell, right? Olfaction is the only sense not to synapse in the thalamus. Also remember, smell was a rapidly adapting sense, right? Once you're in a room with an odor, after a relatively short time, you barely notice the odor anymore. All right, that's it for the review. Now, because I'm a tyrant, and well, not because I'm a tyrant, but mostly because there's not a lot of time, I'm going to go right into the fun activity.